One of my research topics is about exploiting the microbial information to sustain food traceability. We all know how important is the food traceability for safety reasons, but also for ensuring the quality of food that we are eating. There are many studies and many evidences that support that the microbial communities are the relevant actors in fermented foods. Let's say, for example, behind a good wine there is a good microbiome because it participates in the fermentation process and contributes to the bouquet of the wine. And so the microbial signature can be used to identify the wine when the cultivar is the same but the vineyard is different. So there are many evidence now that support this microbial signature in fermented food, but what about the non-fermented foods? Are the microbes important as well? Can we use the microbial signature to trace the origin of non-fermented foods? The answer is yes, of course. It's more difficult, but it's not impossible, it's feasible. And we do that with insect-based novel food. We all know that EFSA authorized the edible insects in our diet. So now there is the great issue of ensuring the traceability of these items, and this is also possible in, for example, for the case of the giant red shrimp, Arisomorpha foliacea, that is present in Mazara del Vallo, and we already know how good and important is this food, but also can be caught in other different geographic area because it's really widespread. So the species is the same, is always Arisomorpha foliacea, but the provenance makes the difference considering also the cost of this food. But we can exploit the microbes associated with the host to trace the geographic origin of this shrimp, so preventing food frauds in this specific case. These are just two case studies, but we can apply this model to other foodstuff and trying to ensure the safety and the quality of the food that we are eating.